plaisir d'accueillir Graham F, que je ne connaissais pas auparavant, et donc j'étais très heureux de faire sa connaissance aussi. Je le connais bien sûr au travers de ses travaux, parce qu'il y a quelque temps, nous avons travaillé sur un phage d'une mycobactérie opportuniste de l'homme, en particulier au cours de la mucoviscidose, et bien entendu, en réalisant ce travail dans le laboratoire, eh bien, nous avons appris à connaître tout l'énorme travail que Graham a réalisé depuis des années sur ces mycobacteriophages. C'est un domaine, de mon point de vue, négligé, en tous les cas, it's a neglected field, at least in medical microbiology, but nevertheless, I think it has to be a very important field of research, both the one you are doing and hopefully some of us will go on in the field of medical microbiology. So thanks again to attend our seminar, to present the seminar today.
Merci. Thank you for the invitation. It's a great pleasure to be here. I have this is my first visit to Marseille, a very beautiful city. I hope to come again. I will apologize that my French is extremely poor, so I will speak in English, and I hope that most things will be clear. And I think there is the video available, maybe with translation perhaps. But if you have questions throughout, in the middle of the seminar, please do raise your hand, and I would be happy to try to explain, to provide clarification. So the title, Exploration and Exploitation of Mycobacteriophages, explains and encapsulates our core interests over the past few years. The idea of exploration, of discovering what these viruses are and learning about their genomics and their diversity, and then taking advantage of that information to develop systems for genetics and perhaps clinical tools for understanding the hosts, the bacterial hosts of these particular viruses. The Mycobacteriophages simply refers to the bacteriophages that infect the mycobacterial hosts, but including Mycobacterium tuberculosis, the causative agent of human TB, and so there is, I think, significant opportunity to develop relevant, clinically and genetically relevant tools. And so this is a main reason for studying these types of bacteriophages. Today, I will talk maybe only just a very little about the exploitation, and I'll talk mostly about the exploration, the discovery of viruses of this type, and then using comparative genomics to try to understand the diversity and the evolutionary mechanisms that give rise to the population of phages that we see. Now, this audience, is probably much more familiar with some of the background than many other audiences are. But I would just like to emphasize that the bacteriophage population, viruses that infect bacterial hosts, is really quite a remarkable population in the sense that it is vast, and there's estimated to be 10 to the 6 to 10 to the 7 phage particles per milliliter of seawater, other water samples, and back of the envelope calculation, there's 10 to the power of 31 phage particles in the biosphere, probably an underestimate, but enough to be confident that bacteriophages likely represent the absolute majority, the numerical majority of all biological forms in the biosphere. So the population is huge. It's also highly dynamic, with an estimated 10 to the power of 23 phage infections per second on a global scale, which means the whole phage population turns over every few days, 4 or 5 days. So it's vast, it's a dynamic population, and it's almost certainly old from the types of studies that you're familiar with, for example, with structural comparisons of capsid and virion structures that suggest very early origins. And so because the population is vast, dynamic, and old, it is clearly highly diverse genetically, and trying to understand that diversity is one of the main questions that we're interested in. And so we would like to be able to better define the diversity of the phage population. We'd like to understand aspects and features of how it has evolved, and we would like to resolve a question as to whether there's discrete populations of phages that are evolving independently, or whether in fact there's a genetic continuum across the spectrum of diversity. There's fundamentally two types of approaches that can be used to address this question. There are perhaps others, but two main approaches. Both important and useful, but have advantages and disadvantages and address slightly different questions. Metagenomic approaches, of which clearly much has been done here in Marseille on this question of metagenomic dissection of viral communities, has the advantage of being able to collect very large amounts of data on viral populations. And as DNA sequencing methods advance, really quite substantial and impressive amounts of data. And for various questions has been an incredibly powerful approach, especially for trying to understand dynamic aspects of the phage populations. A potential disadvantage, however, is that what you get is lots of data, but you only get data. What you have is information that sits in the computer. And it's very hard to associate the viral sequences that you have to specific hosts. The alternative approach is a genome by genome approach, where individual bacteriophages are isolated and sequenced and compared. Obviously, this generates much smaller amounts of data, very small compared with the metagenomic data sets. But the advantage is that you get whole genomes, and you have information that sits in the computer, but you also have resources that sit in the freezer. And so you have entities, organisms that can be propagated, grown, studied, functionally dissected, and analyzed in all sorts of ways. The disadvantage is that to sequence the genome by genome approach, you have to have the phages to sequence, the individual phages to sequence. And it is quite laborious and time-consuming to isolate and purify individual phages. But at least you do have some information about the host, because you know the host that you've used to isolate those phages. And so we needed to choose a host, and our interest in the mycobacteria led us to think about Mycobacterium tuberculosis as a host. Unfortunately, TB grows very slowly, with a doubling time of 24 hours. And so it takes a very long time to grow and to see colonies or phage plaques. And it's a pathogen. So instead, we chose a related strain called Mycobacterium smegmatis, which is relatively fast-growing. It's a non-pathogen and can be used readily to isolate phages from environmental samples. And so the mycobacteriophages that we'll describe all infect Mycobacterium smegmatis, and there is a subset, groups of those that also infect Mycobacterium tuberculosis. Clearly, this is a very small subset of the total phage population. But because they can infect the same host, the exact same host strain, we would predict that they would be in genetic communication with each other, and that we would see that when we compare the genomes and examine their diversity. So how we solve the problem of being able to isolate, purify, make DNA, sequence large numbers of individual phages is by the development of integrated research and education programs. And so I will not give a lot of details, just to say that we have developed, for example, a program called the Phages program, together with the Howard Hughes Medical Institute. And this now offers a course for first-year undergraduate students. And what those students do is to collect samples from the environment, isolate phages that form plaques on Mycobacterium smegmatis, and in fact some related strains, and then to purify those phages, examine them by electron microscopy, extract DNA, sequence the genomes, compare the genomes, and get insights into diversity and evolution. And so now this program is quite large. Last year there was a total of 2,650 students at about 75 universities and colleges, and the program continues to grow. And this year, maybe 100 schools and universities with over 3,000 students. And so this is educationally rich, but also advances the scientific questions about diversity. This is just showing the impact of the programs. There's actually two different education programs on the number of sequenced Mycobacteriophage genomes. And so many years ago, back in 1993, soon after I started at the University of Pittsburgh, it was quite difficult and took a couple of years to sequence the first Mycobacteriophage. And now, this is now zoomed up to, we'll see something like 850 or so complete genomes. And clearly, this is fueled by the simplicity of DNA sequencing as the technology has advanced. But it doesn't matter how fast or how cheap or how simple the sequencing is if you don't have the viruses isolated in the first place to sequence. And so these programs have really helped to advance this data set of phage information. And so we have a database and a website here at phagesdb.org, which is where the information from all of these genomes is collected and made available. This database now actually includes phages from across the phylum of the Actinobacteria, but I'll focus today just on the Mycobacteriophages, mostly on this group of Mycobacterium smegmatis phages. There's now almost 6,000 individual isolates have been recovered and in the freezer. About 860 or so have been sequenced, and that's mostly what I'm going to talk about. So we have GPS coordinates on most of these And you can see that although there's a few that have come from elsewhere in the world, by far the majority are from the United States, and there's lots in Pittsburgh, which is right here, of course, but they are from across the continental United States. And as the program grows and we have more international participants, we hope that we will have a broader geographical isolation of these and related types of phages. This information, incidentally, lets us look at correlations between the geography and the types of phages and types of genomes that are discovered. I won't say too much more about that, but I can answer questions later if anyone is interested. Okay, so what have we learned about these phages? So first of all, just want to mention the general properties and the morphotypes of these phages. So if you just look at the plaques, there's quite a variation, some big plaques and some small plaques, fuzzy, hazy plaques. But all of the phages that we've isolated are double-stranded DNA-tailed phages in the classification of the Cordovaranians. No single-stranded DNA phages, no RNA phages. We don't know whether they just don't exist or whether the methods have excluded their recovery. Moreover, all of the phages are of two morphotypes within this group as viewed by their tail structures. The so-called cyphiviridae with these long flexible tails and the myoviridae with these contractile tails. So most of what we have look like this and some look like this. We don't see any of the podoviruses that have the little short stubby tails. We know relatively little about the detailed structures of these, although I'd like to just give a nod to a very nice reconstruction that was done here by Marcel and colleagues, by Michelle and colleagues, of this particular phage that they recovered from Mycobacterium abscessus. And so, in detail, this probably shares similarity to many of these cyphoviral types, but this is providing a much nicer high-resolution view. If we just look more broadly at a bunch of different types, you can see, as I mentioned, that many of them look very similar to each other. You can see some minor variations in tail length. Here's the myoviruses here with the contractile tails, and many others that look very similar. You might think that they are essentially the same, but they're not. And we have some that have these long non-contractile or flexible tails, but instead of having the soccer ball, the football-shaped capsid, they have these prolate capsids that are long. And this is one of our favorites, which is a phage that's called Corndog, and the students named it because it looks like this American delicacy, which is a hot dog dipped in batter. If you've never seen one of these, you don't have to try one yourself, but this is what they look like. It's called Corndog, and in fact we do have quite a good high-resolution reconstruction of this unusual capsid structure. But that's for the most part what we know about the structures of these viruses. So clearly, to understand genetic diversity, we need to explore these at the genomic level, and just want to start by showing you a broader view, if you like, a 10,000-foot view of how they appear. And so this can be shown in a very simple way by a dot plot, where we simply make an array of genomes, so lots and lots of genomes end to end, here maybe a couple of hundred, and then compare them to themselves. And so wherever genomes are similar, you see a diagonal line. And if you just take a random array of genomes, as shown here, and compare with itself, you can see a random display of similarity amongst pairs of genomes. And then we can use that information to rearrange the position of the genomes in the array so that those that are similar to each other are grouped together. And that simply gives you, it's the same data, but with a different representation, gives you this type of dot plot. And so now there's two very, I think, obvious conclusions, is that a lot of this sequence space here does not show similarity. So there's quite a large number of completely different types that don't share DNA sequence at all. The first conclusion is there's high diversity even amongst these phages. And secondly, that there are nonetheless groups of phages which are much more closely related to each other than they are to everything else. And it's a fairly obvious conclusion. And so, for example, here is a bunch of phages, a group of, 10 or so phages, all of which are very similar to each other, but different from everything else. And you can see these other groups here emerging quite clearly. Some of the groups appear to have some obvious subdivision. And so if you just look at these groups of phages, most of these that have the dark black line are very similar, but then there's this one at the end, which clearly has a different type of relationship. It has a much fainter representation of similarity. So just to acknowledge the landscape of diversity at this level, we've grouped genomes that are similar to each other in what we refer to as clusters, and we just give them a letter, cluster A, B, C, D, E, etc. And then where there are obvious subdivisions, we make them subclusters C1 and C2, or perhaps subclusters B1, B2, B3, B4, etc. And so this is not meant as a strict phylogenetic distinction, but as a taxonomy of convenience when talking about and trying to analyze the genomes. So if we just look at the database to see the current status of what has been isolated, and remember, these are all phages that infect exactly the same strain of the same host. What we see is all of these clusters listed from A down to T. There's 7 that we refer to as singletons because they don't have any relatives, they're just one of a kind. And you can see that many of these can be subdivided, cluster A now into 13, I think maybe now up to 15 divisions of this, whereas others appear to be much more homogeneous, although they have many fewer members. Okay? And so lots and lots of different types, lots of different sequences that are basically completely unrelated to each other. And when we look at the numbers of the members, that's highly biased as well. About half the population belong to this cluster A, 300 or more individual genomes of that general type. But then these other groups have maybe just 1 or 2 or 3, or obviously just 1 for these singletons. So the exact number that have been described doesn't really matter. The more genomes we sequence, the more this diversity continues to expand with new singletons and the creation of new clusters down will soon be, I'm sure. In fact, I think we already have a cluster X to be added to the bottom of this list. So that's an overview of what the phages look like in general. I'll just mention some parameters about genome length. And so they vary from 40 kilobases to 120 160 kilobases. The average is about 70 kilobases. And so each encodes, on average, about 100 or 110 genes, predictive genes. But I want to point out also that if you look at the GC content, even though the host, Mycobacterium smegmatis, has a GC content of 67.3%, the bacteriophage GC content varies quite enormously. The average is quite similar to this, but the distribution is broad and goes from a low of 50.3% GC and a high of about 70% GC. So we can see that the diversity is great, but the span of GC content is also quite large. And we'll return to this because it's a way of getting some insights into the evolutionary mechanisms. So I'm not going to show you a lot of detailed maps, but just as an example. And this is a phage called Patience. It's actually a low GC content phage, and I'll come back to it. But it shows the typical genome structure or architecture with individual genes packing at high density the genome. The structural genes, the genes making the capsid, tail, tend to be grouped in this left part of the genome. The right parts of the genomes tend to have a very large number of small open reading frames, many of them very small, and we have no idea what they do. And in fact, of all of the genes that we've identified or predicted, 100,000 something, maybe only about 30% can you assign functions to or can find homologs in the database from genes that have been sequenced from other sources. And so this is clearly a very large reservoir of genes that for the most part we don't know what they do. And of course, this is true not only for bacteriophages, but it's true for the viral population as a whole. Okay. Turns out that some of the colors are not represented on this slide, which is going to make it hard to see. But I can explain the point I was trying to make, that I want to emphasize that the putting of the phages into clusters is just really an artificial system of convenience. And the clusters themselves vary enormously. Some clusters of phages are all very similar to each other. Other clusters of phages are very different to each other. And so there is no universal description of the diversity within those groups. It's extremely varied. And what is hard to see here, but you can see it on the computer, is that for this particular cluster of phages, these genes here, which are the structural genes, are extremely similar to each other, and there's relatively little variation. But then once you start looking down in the right part of the genome, there's an enormous amount of variation, as though this is a playground of recombination, which is bringing different genes in and out of these types of phages. And so the dominant theme that describes the architecture for phages is that they are pervasively mosaic. They have genomic mosaic structures. And we can think about them exactly the same way that the Romans built their mosaic art. The Romans would use different tiles of different colors and with different geological histories. And so these tiles can be put together in lots of different ways, and you can make lots of different pictures of them. And phage genomes are clearly put together in exactly the same way, with the tiles, as I'll show you, the tiles corresponding often to just single genes. And so the variation in the diversity can be explained in part by the different combinations that you can put the tiles together to make different pictures, with each picture being a different type of phage genome. We don't know how many total motifs or tiles there are, but it's clearly a very large number, and therefore there's a huge variety of ways in which they can be assembled in this way. So mosaicism is the dominant theme, and I'm going to show you a couple of examples that illustrates the mosaicism. We can see the mosaicism at two different levels of analysis. We can see it by DNA sequence comparison, where we can visualize relatively recent evolutionary events. And we can look at the amino acid sequence similarity, where we can look at much more distant events or departures in evolution that have occurred such a long time ago that you no longer see the evidence at the DNA level. So first of all, a couple of examples of DNA sequence mosaicism. So now here at the top I'm showing a dot plot of now just one genome against one other genome. So this is a particular genome, a relatively small one, and this is a second genome, a relatively large one. You see no diagonal line. They're essentially unrelated at the DNA sequence level. But if you look hard, you can see a small region here, which is actually quite closely related when you look more closely. And in fact, the alignment shows you that the DNA level, these are about 95% identical at the nucleotide level. So clearly a gene that has been acquired relatively recently. And I think we can make a good argument that this large myoviral genome has acquired this recently. And it's actually the gene that encodes a repressor, a phage repressor. So it certainly appears as though this phage here has stolen this repressor for some reason that remains a little bit unclear. If we look at the bottom panel, you can see another example where this is actually the same phage on this axis as shown here. And actually now we have a different phage, but also a larger myoviral phage genome. And what you can see is, again, there's no DNA sequence similarity, but here is a small region where, once again, when we blow this up, we can see that it's actually quite a good alignment. The DNA sequence is 97% or something identical. And once again, this is the theft of the repressor. Only it's happened, apparently, in at least two different independent events. So we still don't know why this happens or the biology behind it, but now we have two separate events where the myoviral type of phage genome here has stolen this repressor. So this has happened in two different places. There's no evidence for any DNA sequence homology that's involved in the recombination. And we propose that this, and in fact all of these types of mosaic events, are mediated by non-targeted, random, illegitimate recombination events. They are simply recombining unrelated DNA sequences to form new junctions, new types of sequences that have not been present before. And so this is a highly creative process in terms of generating variation. And although it may happen at a relatively low frequency with the vast population, it's highly dynamic, it's been doing this for a long time. Frequency is not so much a concern about mechanism for generating recombination. And again, I apologize, the colors, some of the colors here have not reproduced on the screen, but I just want to point out that this is a phage that has taken this repressor and by doing the alignment with a related phage in this case that hasn't done it, that lacks it, we can actually see the events that have happened to give that particular type of exchange. So all of that is looking at what's happening at the DNA sequence level. If we look at the amino acid sequence level, we see what is illustrated here in spades. And so this is one example that we can reproduce this for thousands of the genes in the genome. So for example, here's a phage called Barnyard and another phage called Patience. They are unrelated at the DNA sequence level, and yet they share this gene here that clearly has a common ancestor. It's got about 37% amino acid identity, so they're not closely related, but clearly have a common ancestor. And yet this is sitting in a completely different context in the two genomes. The sequences to the left and the sequences to the right are completely different in the two genomes. We suppose this happened through one or a series of illegitimate recombination events, but it has situated this genome with different neighbors in a different context. If we look at Patience in TM4, we can simply step one gene over. This gene has weak but significant similarity with this gene here. And once again, the sequences to the left and the sequences to the right are completely different. It's in a different context. In this phage TM4, we can skip over one gene over. This gene here has about 52% identity at the amino acid sequence level with a gene here in an otherwise unrelated genome called Twister. And again, the genes to the left and the genes to the right are different. It's in a completely different context. So in this case, these are single gene elements, single gene mosaic tiles that are being put together in different contexts, just like that picture of the Roman mosaic that I showed. Okay, so that's what the architecture of the genomes looks like and how they're put together. Now what can we learn about this question of whether there are discrete groups or whether in fact there is a continuum? At first, it's very simple to think that because we have taken some genomes and put them into clusters, it's tempting to think that those clusters represent discrete groups. Some of them do, but the question is whether that's a general property or whether it's simply that you get clusters of genomes because you have a lot of them, not because it is discrete and unable to recombine with the other phages. And so I want to show a couple of representations that I think show quite clearly that these really are not discrete groups in that way. This is a network representation by gene content. So we can simply ask to what extent do the genomes share genes from one to the other and display them using this program splits tree. And you can see that cluster C phages, which are these grouped over here, certainly look as though they have quite a discrete structure to them. They seem to be quite separate from the others, and certainly are not closely related to any of these other types. With the cluster A phages, they also look fairly discrete, although there's a reasonably thick stalk here, which reflects sort of the complexities of the network relationship. But once you start looking elsewhere in this network, you see that there are relationships that are much more complex than that, simply because you have groups of phages that are essentially artificially grouped into particular clusters like N or I or P, but in fact they're sharing very substantial numbers of genes with each other. And we think that is much more consistent with this nature of a continuum of diversity. And so this is the second representation that I want to show you, which is looking at shared gene content as a heat map, So what we've done is to do a pairwise comparison of one genome with another genome and calculate how many genes they're sharing as a percentage of their genome. And if they're very, very similar, that's going to be a red color. And if they're very dissimilar, it's going to be this very pale blue color. And so what we've done is to do that for all of the genomes. So each of those comparisons is a pixel, essentially. And then we've oriented the genomes, again, according to the clusters that they have been designated in by the previous analysis. So we can look then at this display. We can look at how similar the genomes are to each other within a group, and then how similar they are to all of the other phages. So this actually is those cluster C phages. The cluster C phages are up here. You can see they're kind of red, so they're very similar to each other. There's relatively low variation within the group. And in fact, they look quite discrete because you see very little color outside of that. There are some shared genes, but not very many. If we look at a complex group such as the cluster A phages, first of all, you can see that the subclusters emerge very clearly. But again, the nature of the variation is very different. Some of these are very red. Some of these are a mixture of red and yellow colors. So the diversity is different. And you see some similarity, some shared genes with these other phages, although not a great deal. But now when we start to look at this complex series of different types of phages that are occupying the central part of this display here, you can see that there's both a large variation in complexity or diversity within the clusters, but then there's also substantial sequence sharing outside of these groups as well. And so basically everything goes. It depends on what you want to see. You can find groups that are very similar to each other, and you can find groups that are very diverse and highly divergent. And it's much more consistent, we think, with there being a continuum of diversity, but with certain groups being much more prevalent in the data set than others. And it's part of the reason why it's important to dig deeply into the number of genomes. With only a few genomes, this would be very hard to interpret, but with 800 or so genomes, you have the opportunity to discover the rare types which illustrate the complexity of the population. And just very briefly, we can do rarefaction curves on the types of genes that are being extracted and discovered. And one of the somewhat surprising observations is that if you look at this rarefaction curve here, this is the predicted hyperbola, and so this is what it would look like if we predicted out to saturation. In fact, the rarefaction curves from the data show that we don't fit the hyperbola, and in fact this continues to increase, essentially telling us that if we continue to isolate phages, we will continue to identify new phages and continue to identify new genes, apparently forever. And so this is probably reflecting the highly dynamic nature of the population in real time, in timescales that are relevant to the isolation and analysis. So I've shown you that we think we have a spectrum of diversity of these phages. I should say that there are no core genes. There's no core genes equivalent of 16S. But in fact, even though you might think there would be a core gene such as a capsid gene, everything has a capsid gene, but the sequences are so diverse that there is no amino acid sequence element that's in common to all of these phages. There is no sequence of amino acids corresponding to, say, a domain that's in common to all of these phages, even though we are just looking at the phages of one strain of one species of bacterial host. I've told you that there's lots of small genes, and most of these genes we don't know what they do. So we have a model for thinking about how the diversity is generated, which is based upon really two main sort of aspects. So here we have our host Mycobacterium smegmatis in the center. This strain is called MC²155. And we can imagine a blue phage and a red phage representing just different particles, different sequences. And we would argue that these phages are migrating across the diverse bacterial landscape rapidly, moving from one host to another bacterial host, where we happen to have captured them, in this case at Mycobacterium smegmatis here, but we would argue that they've come from very different places across the microbial landscape, moving at high frequency from one strain to another, and provided that the diversity of the bacteria are present, the bacteria essentially act as stepping stones for the phage to jump from one host to another. And as they do so, they sample different parts of the core gene pool, and so they can pick up and exchange and acquire and swap and donate different sets of genes depending on the pathways that they've taken. And essentially these parts of host dynamics is really what is determining their genome diversity. And so for a phage like Patience, which has a low GC content, we would argue that it's likely come from a place that maybe had a relatively low GC content series of hosts, and then has relatively recently found its way into the high GC Mycobacteria. And I want to briefly show you a little bit of evidence that I think supports that hypothesis. So here again is our phage Patience that I introduced earlier with its low GC content. It's just impossible to see, but I'll explain that perhaps not surprisingly, because the GC content is so different, the codon usage profile is also very different from its host. And it's different in remarkable ways. For example, there are codons that are extremely rare in the host. They have to be read by wobble-based pairing, and those codons in the host are extremely rare. But in Patience, those codons are the most commonly used codon. It's a total misfit in terms of codon usage. Surprising for a phage, which you would think wants to reproduce in a very efficient way, and which it clearly does. One possible resolution could be that Patience has picked up tRNAs to accommodate this different codon usage. And although it has one tRNA, it clearly cannot account for this phenomenon. So one of the things that we've done is to, first of all, ask whether we can detect those phage proteins being made by a proteomic strategy using liquid chromatography tandem mass spectrometry. And the bottom line is that we can, with a red dot, have just indicated all of the products that we can identify. So this includes many of these small open reading frames that I mentioned to you. So they are not annotation errors or annotation over exuberance. They are really being made. And we can use that information to find out or to interpret levels of expression. In other words, which genes are being highly expressed and which genes are being expressed at lower levels. And then we can do a kind of analysis where we compare the variation in the codon usage for individual genes with how highly expressed they are, and we see a good correlation. And when we further dissect this variation in codon usage, we can see that genes that are highly expressed are moving their codon usage towards that of Mycobacterium smegmatis, whereas other genes that are expressed at lower levels haven't done so. And so this is exactly the profile that we'd expect for a phage that has come from a low GC environment and has recently entered the high GC environment and is in the process of adapting to be able to express its genes in that host. And so this is probably reflecting what is going on a broader scale. It's just that the extreme of the GC content enables us to do that analysis. And so at a final point I wanted to make, which was that I said I wouldn't talk a lot about tools, genetic tools, but in brief, one of those tools we've developed is a recombinating system for the mycobacteria and for Mycobacterium tuberculosis. So we can express these two phage recombinase genes. One's an exonuclease, one is a DNA pairing enzyme. And then we can get mycobacteria to do efficient recombination, either exchanging a double-stranded DNA substrate to recombine with a chromosome, or a single-stranded DNA oligonucleotide substrate to put a single base pair mutation into the chromosome. And these recombinating tools are quite broadly used for TB genetics, for construction of mutants. I mention that because we have a particular application of that, which is the ability to construct mutations in the phages themselves. And so this is an important tool for us. It is called BREAD, or Bacteriophage Recombinating of Electroporated DNA. Because now we have the tools to go into the genome and ask which genes are required for growth, and to make mutants that will let us dissect how the phages work. And this is obviously the advantage of the genome by genome approach, that we have all of these genomes in a freezer, we can pull them out, we can grow them. Now we have the genetic tools to construct mutations in them. So we've been doing this quite a lot. I'll just give you one brief example of a phage which is called Giles, which was named after the English cricketer, apparently. And what was done here was to take each of the open reading frames of the non-structural genes. So these are the structural genes. We know that they're essential, so we did not mutate them. But here we've gone through and attempted to knock out with a deletion mutant each of these genes. And the ones in yellow are genes that we can, in fact, knock out and show that are not required, are not essential for lytic growth, for forming a plaque. The green genes we know are essential, and the blue ones we think are essential, but are less confident. And so now we have the tools to make any mutants that we want in these phages, and then to determine particular functions. So, for example, we show that a couple of these genes are indeed involved in DNA replication, roles that we didn't previously know about. Okay, so I started out with some questions, and I think that we have at least some answers. I've shown you that we can define this diversity of phages for a single host strain. Now we're starting to look at phages with large groups of phages of related strains in order to understand the second dimension of the diversity. I've shown that this has evolved through the mechanism of illegitimate recombination to make these mosaic genomes, and proposed that migration of phages across the host landscape is what gives rise to this diversity. And finally, I think that we have compelling evidence that we really have a continuum of diversity, and that continuum of diversity will continue to be expanded upon and filled in as we begin to isolate and study many more phages of this phylum of bacteria. So let me give brief thank you to my faculty colleagues, Roger Hendricks and Jeffrey Lawrence, who have participated in much of this work. A lot of the genomics has been done by Dan Russell and Welkin Pope, together with Charlie Bowman, Steve Creasorn. The recombination work was done by Laura Marinelli and Rebecca Diedrich and Julie Van Kessel. These are my colleagues at the Howard Hughes Medical Institute that have helped to develop the education programs that I mentioned. My colleague Bill Jacobs in New York. Several thousand, perhaps 6,000 student phage hunters everywhere. Thank you. And for funding from NIH and from the Howard Hughes Medical Institute. And I'd be delighted to take any questions.
The problem with our talk is that we have to sit down for the entire afternoon in order.
Yeah.
But we have a few minutes. So, alors, des questions?
Yes, Graham. Do you think that a general strategy to isolate phages against with hosts that are difficult to grow in laboratories is the approach that you used, like isolating TB phages on smegmatics? And for example, what is the proportion of phages isolated on smegmatics that infect TB?
Okay, so so first the general methods for isolation, as far as we can see, can be broadly applied to other bacterial hosts. We've focused on the Actinobacteria, but yes, we've had some hosts appear to be a little bit more productive than others. But in terms of, I'm sorry, the second question was about
the proportion of
the proportion of phages that infect TB. Okay, so the phages. So we've only looked at one strain of TB, and we're very interested in looking at a large range of TB clinical isolates. So we're gearing up for that. But using just the one lab isolate of TB that we've used, the phages that infect it are of a particular genomic type. And so, for example, all of the phages in cluster K and in all of the subclusters all infect TB that we've tested. If you look in cluster A, most of them don't. But essentially all the phages in subclusters A2 and A3 do infect TB. So there's a very strong correlation between genomic type and the ability to infect TB. As far as we can tell, although it's a preliminary analysis, we think that most of that distinction is at the surface level, at the level of absorption, because at least for some of the phages that don't infect TB, we can electroporate the DNA into TB and have it propagate and make phage particles that we can then amplify in the assay using Mycobacterium smegmatis. So we think that for most of the phages, it's not an internal metabolic or an expression problem, it's an absorption.
I have a question. Do you think that is there any organized mechanism to favor the exchange or the recombination between genomes? Do you think there's just a random process? Because the way you showed it, it could be that, well, by natural selection, complete orths are selected, but it looks like there is a more organized mechanism to favor the mosaicism.
For the examples that I've shown and we've looked, we see no evidence for homology that's generated mosaicism. Although I must emphasize that I don't want to say that homologous recombination does not happen. It does, and it's very important for rearranging combinations of genes. But homologous recombination doesn't generate new boundaries between genes. per se. At least that's not what we see here. And so we think that it's random with natural selection for function. And the question is it mechanistically driven in some senses? And there's not a lot of information on it, but there is some data that suggests that some of the phage-encoded recombinases are much more willing to recombine short and often weakly related sequences. And so one could imagine a particular role that phage-encoded recombinases play, which mechanistically catalyze recombination, but do so in, let's just say, sloppy kinds of ways.
What about places?
What about places?
Nucleases.
Or nucleases. Oh yes, and I apologize, I mean there's a lot of other mechanisms. Clearly transposition, HNH and nucleases, things that are cleaving the DNA and then rendering it subject to repair. I mean CRISPRs and other nuclease systems that are cleaving DNA are all, we think, likely to promote essentially recombination. And we have some evidence for that for some of the HNH and the nucleases. The transposition will do the same thing as well. Whether they're generally responsible for all of the mosaicism seen more broadly isn't clear.
Is there any significant relationship between the GC content and some phenotypic features of these phages regarding the size of the genome, size of particles?
A long time ago, we had looked at the relationship between genome length and GC content amongst all bacteriophages and saw a positive correlation. We haven't gone back and done that analysis with what are now much larger data sets. I wouldn't be surprised if it falls apart somewhat. For these phages, there's no obvious phenotypes that are associated with, for example, with variations in GC content. Patience, for example, appears to grow perfectly well. It grows to high titer, makes normal-looking plaques. It's indistinguishable from any of the other phages that have higher titers in anything that we've been able to look at. And I should mention this GC content. I mean, we see it here with this group of phages. I mean, this is the largest group of phages for any single host that's been sequenced. But in general, if you look at groups of phages by host, in many other cases, you also see this very high variation in GC content as well. And so we've been looking at phages of other high GC actinobacteria. We have genomes in about 40% or something. So it's a big range. And there's not any obvious consequence to it, at least as far as we can tell.
I was wondering if this wealth of genomic data helps us understand how the viruses, the phages, go through the thick wall of the bacteria, and if there is a diversity in there as well.
Okay, so the question was, and there's a second part of the question, how do the phages inject their DNA into the mycobacteria, given the thick mycolic acid-rich cell wall and the mycolic acid as an outer membrane? And then how do they get back out again as well? We have some information as to how they get back out. They use enzymes that specifically cleave the linkage of the mycolic acids from the peptidoglycan. Those enzymes they're a set of esterases. They're not found in all of the phages, but there are most of them. If you knock them out, you simply form smaller plaques with a compromise in production. The phenotype is similar to what you see for spanning mutants in phages of gram-negative bacteria. In terms of how they get in, we know very little about any of the receptors for these phages specifically. We're starting to address it but it remains a bit of a mystery as to how they get through these, through the structures. There are certainly some of the phages that have tail proteins that appear to have enzymatic activities that are associated with them. And so we're very interested in trying to dissect those. But the ones that are most clearly recognizable are things that degrade or remodel peptidoglycan. And so that doesn't really answer the question as to how they manage to get through the other components of the cell wall. That's great questions, and we're hoping to learn more about it.
My first question is, because of this project, have you observed any incidents of gene capturing by the Mycobacterium from the host? Number one. And secondly, how close is this kind of project to phage therapy, like the use of phages in treating Mycobacterium?
So let me answer this last one first and see if I can remember the first one afterwards. In terms of phage therapy for TB, so I would argue that there's a need for phages. There's a need for new therapies because of the rapid development of multi-extensively drug-resistant strains of TB. I suspect, and I agree with those who have argued, that phage therapy alone is unlikely to be a substitute for antibiotic treatment. But I think that there is a potential role for the inclusion of phages in a therapeutic regimen with a view to both interfering with transmission, which I think may be the most important role, or alternatively for reducing the incidence of multi-drug resistant and XDR strains. And so we're interested in that and are in the process of trying to sort out the particular phages that we have that infect TB, to look at panels of clinical isolates to see their range, and to particularly define the receptors and the mechanism of resistance with a view to generating a cocktail of phages that will contain 3 or 4 types with different resistant profiles for each of the different phage types of clinical isolates. There's a lot of work to be done to going from where we're at to having a cocktail that could be tested for therapy, but I think it's worthwhile doing. And the first question was?
Gene capturing.
The question is whether there's obvious capture of genes from the host. So, I mean, I think there are genes that I would argue were obviously captured from the host, but when it happened and from what host is unclear. You know, there's quite a smattering of genes which most people would probably not refer to as phage genes because you find them in bacterial genomes, not in prophages, but things like LSR2 is a good one. There's things to do with quercetin biosynthesis, there's some toxin genes, there's a whole variety of things. Clearly, they look like phage genes to us, but where and when they got them is a little bit unclear. That for the most part, you don't see very high degrees of sequence similarity with Mycobacterium smegmatis. Okay? But that doesn't surprise us because we don't think of the phages as really being smegmatis phages. I mean, they are, but they've evolved through relatively recent evolutionary time through other hosts. And so, for the most part, we don't see these really close similarities between phage genes and host genes that would suggest that they had been acquired very, very recently in evolutionary time. So maybe one more.
So they are all lytic phages, or we have the genic one?
So some of the phages form clear plaques and don't form lysogens. Some of the phages clearly do form lysogens, but they do so at a broad range of frequencies. So to the extent that looking at classifying by plaque type is a little bit hazardous. It's further complicated by the fact that the phages that form clear plaques, completely clear plaques, very evident, look like a classic lytic phage. in pretty much in every case, those phages are mutants of a temperate parent. About half or so of the phages, or half of the groups of phages, encode either an integrase or a partitioning system, which are characteristics of temperate phages. And so there's a very large number of temperate phages present, and in different types. There are phages that we're pretty sure don't form lysogens, but form hazy plaques. And there's still groups of phages for which it's unclear whether they really form stable lysogens at low frequency, or they just don't kill very efficiently. And so it's a rather a grey type of scenario as opposed to a black and white classification.
Okay, I think we have unfortunately to stop there. Thanks so much again.